work regarding toxic astrogliosis, we started back almost like 3 or 4 years back. So first we started to develop in vitro models, in vitro CNS cell-specific models. So we have cultured primary human astrocytes, primary human neurons, primary human oligodendrocytes, and microglia, and try to collect the supernatant from these cells and identify which biomarkers are present there, like which proteins are released from these cells. And then we try to identify the clusters of these biomarkers by running the variable cluster analysis. So it's a statistical method. And we found the clusters which are clusters of the biomarkers. Basically, it's the logic behind that, the proteins which are released from the specific cells, if they behave same in a disease or in a healthy patient, they should give a cluster. So then we got these clusters, and then we went back to our MS patients, and we have seen like which clusters get elevated in MS patients and which clusters strongly correlated with MS disease severity. And we were expecting that it will be more like neuronal clusters or oligodendrocyte clusters, because generally neurodegeneration or oligodendrocyte loss is considered as a hallmark of MS disease. But we found astrocytic cluster, like one of the astrocyte cluster was strongly correlated with disease severity, MS disease severity, and it was elevated in MS patients. And at the same time, there was a paper published in Nature, which has talked about the A1 astrocytes, and they coined the term A1 astrocytes for the toxic. We were using the term toxic astrocytes. They use the term A1 astrocytes. And they have shown these A1 astrocytes, they are even present in MS lesions, and we were showing the same thing. And even they have shown these A1 astrocytes are present in different neurological disorders also. And they have identified C3, complement component 3, as one of the marker of this A1 astrocyte. And we also have seen that C3 as one of the protein present in our cluster. So we started to think about this, our study as well as study by this other group. It has shown the role of this toxic astrocytes in MS. So we did drug library screening to identify the drugs which can block the induction of toxic astrocytes. So basically, in physiological level, or in physiological brain, astrocytes are quite helpful for the functioning of neurons. Like they play the supportive role. But during the disease condition or neuroinflammatory conditions, the astrocytes get converted into toxic astrocytes. The process is called toxic astrogliosis. And once they are toxic or A1 astrocytes, they cause some neurodegenerative activities. So we were trying to look the pathways which play a role in this toxic astrogliosis or induction of toxic astrocytes. And for that, we use the drug library screening. So we use a commercially available drug library, which have almost over 1,400 drugs. And our logic behind using the drug library is stop using the single drugs, because this drug library have the multiple drugs with the similar targets. So if we see those group of those drugs blocking the induction of toxic astrogliosis, it will validate our finding at multiple levels that this pathway is playing a role in the induction of toxic astrogliosis. And when we ran the drug library screening, we identify mainly drugs related to unfolded protein response, the drugs which can block proteosomes, drugs which can block heat shock proteins, can block the induction of toxic astrogliosis. And it's quite evident that during this toxic astrogliosis, astrocytes they produce a lot of proteins and they secrete a lot of proteins. So the lot of protein production is going on in the astrocytes, and maybe they are under the unfolded. And when a lot of protein production is going on, there will be a lot of unprotein. There will be a lot of misfolded proteins. So maybe astrocytes have the unfolded protein response, or they're under stress of this unfolded protein response. And when we use some drugs which can block this unfolded protein response, they completely block the protein production. And maybe that's why the markers that we were seeing, like complement component 3, the levels of that marker in the cell supernatant was reduced. But when you think at the treatment level, if we use these drugs for the long time, it may kill the astrocytes, because unfolded protein response is is good for the cells, like because cells are trying to survive from the unfolded protein response. So we should have some drugs which can elevate this unfolded protein response, or like help the cells to work better in this unfolded protein response. And one of the drugs that we have identified through this drug library screening, Dantrolene, which is a ryanodine receptor inhibitor. So ryanodine receptors are present on endoplasmic reticulum, and they help in calcium release from ER into the cytoplasm. And it is a FDA-approved drug for spasticity. And we have identified that drug can block induction of toxic astrogliosis. And this drug library screening we have ran at in vivo achievable concentration. Because many in vitro studies, they use very high concentration of the drugs and they show the effect of the drug. But when the drug moved to the preclinical or clinical studies, we don't see similar effect. So to start with, we have used the concentration which can be achieved in vivo. And we found this Dantrolene at in vivo achievable concentration can induce, can block the induction of toxic astrogliosis. And currently, we are using this drug in our ongoing clinical trial. It's a TRAP-MS clinical trial. And this drug will be used only for the patients which show the biomarkers for toxic astrogliosis or induction of toxic astrogliosis. And then we came back to these biomarkers. Like previous studies, which was published in Nature, they have used complement component 3 or C3 as a biomarker of toxic astrocytes. When you think of just astrocytes and toxic astrocytes, definitely C3 is released only by toxic astrocytes or A1 astrocytes. But when you think about whole human body, C3 is released by some other cells also, such as like microglia, monocytes, and so on. Then if we're measuring elevated C3 levels in CSF, then how you can say this all coming from astrocytes? It may be coming from these other cells. So when you are moving in vivo, you should have a biomarker which should be highly specific, because we are using a targeted delivery, which is a targeted treatment, the treatment which is targeting just specific cell type. So we should have biomarkers for that specific cell type. And through our analysis, we have found serpin-A3, serine protease inhibitor A3, is a biomarker which is specific to the toxic astrocytes. Like compared to normal astrocytes to A1 astrocytes, it's released by toxic astrocytes, same like C3 or complement component 3. And even the paper which was published in Nature, they have identified this serpin-A3 as a biomarker of toxic astrocytes, but they use C3 as for their studies further. And when we went back to Human Protein Atlas, as well as the publicly available databases, we found this serpin-A3, at least in CNS, is highly specific to astrocytes. None of the CNS cells, such as neurons, microglia, even immune cells, they don't release serpin-A3. Only astrocytes release serpin-A3. So if we are working in CSF, it's a specific biomarkers for toxic astrocytes. When you think about whole human body, definitely serpin-A3 is released by other organs such as liver, or it's released by especially hepatocytes. So to avoid the problem of this crossing of serpin-A3 from serum to the CSF, we have done something called serpin-A3 index, which is kind of analogous to IgG index. So where we have studied serpin-A3 levels in serum versus CSF, at the same time, albumin levels in serum versus CSF. And we say analogous to IgG index or antibody index, we have looked for the serpin-A3 index, which will look specifically intrathecal production of serpin-A3 or specific production of serpin-A3 in CSF only. And based on that, we see the similar thing. This serpin-A3 is highly specific to toxic astrocytes, and it gets elevated in MS patients and strongly correlated with MS disease severity, which is statistically significant. Correlation of R square was around 0.2 to 0.3. Yeah, it's at an individual protein level, it's low, 20% is quite low. But you can think about MS is a kind of heterogeneous disease, many cells playing role in that. And if a single protein can tell you 20% about the disease severity, it's a kind of a very good marker for disease like MS. So we have identified drug which can be used we are already using in clinical trial, which is a specific to toxic astrogliosis. And we have identified a biomarker which is specific to toxic astrogliosis processes. So hopefully we'll move forward in this work and we'll get some positive results.